So, B lymphocytes. All lymphocytes have a round shape with a round nucleus. And this nucleus is so large that it occupies most of the cytoplasm. They are small cells, 7 to 8 micrometer in diameter. And we can notice that all lymphocytes are small in size because they are not going to undergo phagocytosis. They are not cells of non-specific immunity. B lymphocytes or B cells, they are cells of specific immunity, and thus they should be a receptor. The receptor of a B cell is called B cell receptor or BCR. It is also called membrane antibody, surface antibody, membrane immunoglobulin, and surface immunoglobulin. Immunoglobulin can be abbreviated as capital I small g. Immunoglobulin is also called the antibody, or it is the antibody. Those BCRs are called membrane antibody or membrane immunoglobulins because they have the shape of an antibody. They have a Y-shaped receptor with two antigen binding sites so that they can bind directly to the epitopes of the antigen, to the virus, to the fungi, or to the bacteria of any antigen. So, as we've said before, they possess an antigen receptor called B cell receptor or BCR. This receptor has the shape of an antibody. It is also called surface immunoglobulin or membrane immunoglobulin. But how a BCR works, or how can it defend the body? Notice that every B cell, in order to work, it needs two conditions. The first condition, it should bind to the antigens of the bacteria or the virus or the fungi, the antigenic determinants of the antigens. So it is going to bind through its BCR to the bacterial epitopes or antigenic determinants. And after binding to them, it should receive a signal from the Th cell. This signal will lead to its proliferation and multiplication through successive mitotic divisions. Then it is going to differentiate into a cell called a plasma cell or plasmacyte that will secrete free antibodies in the serum. Notice that these antibodies will have the same shape as those on its membrane. That's to say, the free antibodies secreted by a plasma cell has the same shape and the same antigen specificity as those connected to its surface. Then those antibodies are going to bind freely in the serum or in the plasma to the antigens that has invaded the body.